Hello, we are Hannah Fott
And Malin Scott.
from the North Central Institute of Science and Technology, and we conducted research on the bioremediation of PCBs and the change in bacteria consortiums. PCBs are man-made chemicals that are persistent and ubiquitous in nature. They are the last to be degraded in the environment and can be harmful to humans. In our community, members of the Spokane tribe, whose diet includes fish from the Spokane River, have experienced health issues from consuming high amounts of PCBs. Currently, PCB-contaminated soil is transported to another site, which is costly and disperses PCBs. We hope to find a more efficient way of waste removal through bioremediation to solve this local issue.
The focus of this project is to observe how bacterial consortiums change in PCB contaminated soil over the course of the bioremediation process, and to identify if there are relationships between certain species of bacteria and bioremediation efficiency. In the pan with fertilizer and inoculant, it is predicted there will be lower PCB levels and higher bacteria species diversity. Previous studies have demonstrated that bacteria from the genera Rhodococcus and Corynebacterium can degrade PCB and hydrocarbon compounds. Bacteria species were identified via 16S nanopore sequencing. PCB levels, bacteria consortium, and species diversity were analyzed.
PCB levels were measured over 4 months to determine if bioremediation occurred. After 1 month, a significant decrease in PCBs in both pans was observed. By January, Pan B PCB levels further decreased, but Pan C levels increased. As PCB levels decreased, some genera of bacteria increased within the pans, potentially representing bacteria bioremediation capabilities. Rhodanobacter saw a significant increase in both pans and was the most abundant genus after the first month of bioremediation. The most abundant species in the pans is a known degrader of a neurotoxin with a similar structure to PCBs. Clostridium also saw a steady increase in percent abundance as the bioremediation process occurred in both pans. It has been found that some species of Nitrospira possess a chlorite-degrading enzyme gene which could potentially aid in the bioremediation of PCBs.
Using Simpson's index, diversity levels slightly decreased over time from the control. Using Shannon's index, diversity levels on average increased from the control. This is indicative of overall increased diversity.
Although some genera of bacteria appear to have a connection with bioremediation, we cannot conclude which specific genera of bacteria was responsible for the degradation of PCBs. The nitrogen inoculant appeared to have no significant effect on bacteria diversity or bioremediation efficiency. In the coming months, we hope to evaluate consortium of native fungi and their bioremediation capabilities. Next year, in collaboration with the Spokane Lands Council, we hope to pitch our research to the City of Spokane and look at bioremediation on a larger scale.
We would like to express our gratitude to the following people. Thank you for your time.